Good morning, students. Today's session is about the evolution of databases for omics studies and pharmacogenomics in drug discovery. By the end of this session, you will have a clear understanding of how omics databases and pharmacogenomics are shaping modern research and healthcare. So, let's start. To give you an idea of what we will cover today, here's the outline. We will begin with an introduction to omics and pharmacogenomics, then explore the types of omics data and their significance. Next, we will trace the evolution of omics databases over the years. We will look at the pharmacogenomic databases that guide personalized medicine, discuss their role in drug discovery, and finally, we will address the challenges and future directions in these fields. Keep this structure in mind as we move forward. Let's start with some definitions. So, what is omics studies? Omics studies refers to the large-scale analysis of biological systems, fields like genomics, proteomics, and metabolomics, aiming to understand molecular mechanisms and interactions holistically. And pharmacogenomics, on the other hand, studies how genetic makeup affects drug responses, paving the way for personalized treatments tailored to individual needs. These two fields are closely intertwined and crucial for advancing research and healthcare. Keep this definition in mind as we move forward. Now, let's talk about the types of omics data focusing on genomics and transcriptomics. Starting with genomics, it has two types, which is DNA sequencing and genome-wide association studies. DNA sequencing helps us to decode the order of nucleotides in DNA. For example, DNA sequencing was critical in the Human Genome Project, which mapped all human genes, providing a foundation for understanding genetic diseases. Genome-wide association studies analyze genetic differences across large populations. A real-world example is the identification of genetic risk factors for type 2 diabetes using GWAS, leading to the discovery of TCF7L2 gene, which plays a key role in the disease. Now on to transcriptomics. It has three types: single-cell transcriptomics, gene expression profiling, and RNA sequencing. Single cell transcriptomics lets us study gene expression at the cellular level. For instance, it has been used to study cancer heterogeneity, revealing why some cancer cells respond to treatment while others resist. Gene expression profiling identifies which genes are active in different conditions. For example, it has been used to study how specific genes respond to chemotherapy drugs, helping design more effective treatments. RNA sequencing is used to study transcriptome. A great example is using RNA sequencing to understand how the COVID-19 virus interacts with human cells, helping researchers to identify potential therapeutic targets. These examples shows how genomics and transcriptomics contributes to real-world discoveries, making them invaluable tools for research and medicine. Next, let's look at how mixed databases have evolved over time. First generation databases include GenBank and SwissProt. GenBank stores nucleotide sequences and has been used globally for fundamental research. For instance, GenBank was instrumental in identifying SARS-CoV-2 genome early during COVID-19 pandemic. SwissProt provides curated protein information such as functions of enzymes and how they interact in biological systems. Second generation databases include Ensemble and RefSeq. Ensemble offers genome annotations and comparative genomics. For example, researchers studying genetic disorders like Huntington disease rely on Ensemble to map and compare genes across species. RefSeq provides standardized reference sequences often used in clinical diagnostics for diseases like cystic fibrosis. Third generation databases include TCGA, HMP, and GTEX. TCGA is a cancer genome atlas which helps to identify genetic mutations in cancers such as those in lungs and breast cancer, enabling the development of targeted therapies. HMP is nothing but a human microbiome project which explores the relationship between the microbiomes and diseases. For example, HMP data has been used to link gut microbes to mental health conditions like depression. GTEX, which is genotype tissue expression, studies gene expression across tissues, such as identifying genes linked to heart diseases or organ specific cancers. These highlights how databases have evolved from simple data storage to driving breakthrough in personalized medicine and beyond. Let's now dive into pharmacogenomics databases which are key to advancing personalized medicine. The first database we are going to explore is the PharmGKB database, which collects data on how genetic variations influence drug response. For example, it provides information on how variations in CYP2C19 gene affects the response to clopidogrel, a drug used to prevent strokes. The second database is PharmVAR, which standardizes information on genetic variations which affects drug metabolism. For instance, PharmVAR includes variants of CYP2D6 gene which influences how the body metabolizes antidepressants like Prozac. The third database is CPIC, which is Clinical Pharmacogenetics Implementation Consortium, which provides guidelines for using genetic test results in clinical practice. For example, CPIC offers guidance on warfarin dosing based on the genetic variations in CYP2C9 and BKORC1 genes, reducing the risk to adverse effects. These databases are the backbone of precision medicine, ensuring the drugs are both safe and effective for individual patients. Pharmacogenomics has a transformative role in drug discovery, particularly in two areas, which is drug target identification and validation and personalized medicine. Biomarker discovery, for example, the discovery of HER2 protein as a biomarker in breast cancer has led to the development of targeted therapies like Herceptin, which significantly improves patient outcomes. Pathway analysis has been used to study the mechanism of Alzheimer's disease, helping to identify potential drug targets like amyloid beta proteins. By tailoring drug regimens to genetic profiles, we can optimize treatments. A great example is HLA-B5701 testing, which ensures that patients prescribed the HIV drug abacavir don't experience severe allergic reactions. Genetic testing for CYP2C9 gene helps to determine the correct dose of warfarin, reducing the risk of excessive bleeding. These applications demonstrate how pharmacogenomics is revolutionizing drug development and healthcare. Despite these advancements, there are challenges, which are data integration. Combining data from different sources like genomics and clinical records is highly complex. As for standardization, varying formats in genetic testing data can delay research progress. As for ethical concerns, issues like privacy and misuse of genetic data remain significant barriers to implementation. Addressing these challenges is critical for realizing the full potential of these fields. The future holds immense promise for pharmacogenomics in the field of AI and machine learning, increased data sharing, and expansion of multi-omics data. Tools like AlphaFold, which predicts protein structures, are already transforming drug discovery. Collaborative initiatives like COVID-19 Genomics UK Consortium highlights the power of global data sharing. Integration of genomics, transcriptomics, transcriptomics, and proteomics will provide a more holistic view of diseases, enabling the breakthroughs like new therapies for rare genetic disorders. These advancements are paving the way for a new era in the precision medicine and global health. Thank you for your attention. I hope this session have given you a valuable insights into the evolution of omics databases and pharmacogenomics.